This video is going to cover section 4.4 and 4.5, and we're continuing our exploration of DNA structure and gene expression. Now, in this particular section, we're going to look at gene mutations and cancer. And so a gene mutation, this is a permanent change in the DNA sequence, and germline mutations occur in sex cells and can be passed on to future generations. And somatic mutations, they occur in body cells and are not passed on to future generations. So both types of mutations may lead to the development of cancer. So some of the causes of mutations could be errors in replication, which are mistakes made while the DNA is copied. And these are quite rare, and there are built-in systems which take care of any mistakes that may happen in DNA replication. So more often, mutations occur through mutagens, which can be environmental factors such as radiation, X-rays, and some chemicals, and these can cause mutations in DNA. Transposons are DNA sequences that move within and between chromosomes, and transposons can jump into another gene, thus causing a change in gene expression. And there's an example of that at the bottom of this slide. So the effect of mutations on protein activity, gene mutations, they can have a range of possible effects on protein activity, from virtually no effect really to complete inactivity or even lack of protein production at all. And a point mutation is a single nucleotide change, and it can cause either no change in amino acid sequence or a change in amino acid sequence that produces a protein that does not function properly, or the introduction of a stop codon, which thus shortens the protein. So with point mutations, here is an example of that, and here you can see the point mutations in hemoglobin, and as mentioned, the effect of a point mutation, it can vary a lot. So starting at the top, we've got the normal sequence of bases in hemoglobin. The next one, where you do have a point mutation, that one virtually has no effect. The next one, due to a base change, so the DNA now codes for valine instead of glutamic acid, and the result is that normal red blood cells, in figure B, they can become sickle-shaped, and in C, the base change will cause DNA to code for termination, and the protein will be incomplete. And so this results in profound change in the red blood cells, and of course their effectiveness in being able to do the function that they normally do, which is to distribute oxygen throughout the body. So this is an example where, and just like almost any condition, in some instances there's virtually no noticeable problem, but in other situations can have a profound effect on the functioning of many important functions in the body. So non-functional proteins, frameshift mutations, they involve one or more nucleotides being added or deleted, and this can cause a change in codons that are translated and production of a non-functional protein. So if the codons make a sentence, an example might be this particular sentence here, and so you can see the profound effect that would occur if you deleted just one of the letters, hence the analogy being if you deleted one of the amino acids in the sequence, in the case of the sentence, you delete one letter and you have a meaningless sentence. And similarly with protons, if you delete one amino acid, then basically you will have a non-functioning protein. So that can have a dramatic effect on the phenotype or the characteristics and functions of many things within the body. So here's one example here, where many reactions, they occur in a series, which is referred to as a pathway. And so if one of the enzymes, and again, enzymes largely being proteins, if one of them is non-functional, then it can shut down the entire pathway, which could have profound effects on the person. So mutations, they can also cause cancer, and the development of cancer involves a series of accumulating mutations, which depend on the type of cancer. Now, most cancers follow a common progression. So they often begin as a benign growth of abnormal cells, and then they can become a malignant tumor and spread to other areas. And here's an example of that, where you have, say, some epithelial cells here, where the primary tumor begins as a mass of cells that are proliferating too quickly, and then some of those cells then become cancerous, and they begin to grow out of control and also invade other tissues, and then can spread to other areas through the bloodstream. And that's what are referred to as metastases, when they spread to other areas from the primary tumor site. So from there then, if I get my slide to change here. So characteristics of cancer cells, the primary characteristics are that they are genetically unstable, and so tumor cells, they have multiple mutations and can have chromosomal changes. Cancer cells, they do not correctly regulate the cell cycle, so the rate of division and the number of cells increases at an abnormally high rate. Cancer cells, they can escape the signals for cell death. There are many cells in the body that are regularly programmed to just simply die, and of course they would be replaced by other cells through mitosis. But these ones, they somehow escape the signal for cell death, and they do not undergo that process. So once again, cells will proliferate uncontrollably. They can survive and proliferate elsewhere in the body, actually. So invasion of new tissues, they can occur, and also includes new blood vessel formation, which is another very interesting characteristic of these cells, is that it actually causes the growth of new blood vessels to supply the oxygen and other things that these cells need, and so you actually get a growth of new blood vessels that were never there before the tumor started to grow. So that covers our first basic look at gene mutations and their relation to cancer. So there's a summary of things that we've just looked at and the key concepts there. So we'll now just have a brief look at DNA cloning. So genetic engineering involves altering the genome or genetic material of an organism. And this often involves gene cloning, which is the production of copies of a gene. So gene cloning is done in order to study what biological functions a gene is associated with, to produce large quantities of protein to produce transgenic organisms, or also to help cure human diseases. So gene cloning involves introducing a gene into what's referred to as a vector, often which is a plasmid, to produce recombinant DNA, also just simply referred to as rDNA. So a restriction enzyme, it cleaves the vector and the gene, which combine by base pairing between what are referred to as the sticky ends. So many restriction enzymes, they leave overhangs of nucleotides when they cut DNA, which are called sticky ends because they can easily base pair with other overhangs. And there's some diagrammatic representations of those. And so DNA ligase then, that enzyme, it seals the gene and vector DNA. So the rDNA then is added to an organism such as bacteria, which makes many copies of the particular gene. So here's so cloning in a human gene. Human DNA and bacterial plasmid DNA are cleaved by a specific type of restriction enzyme. And for example, human DNA containing the insulin gene is spliced into a plasmid by the enzyme DNA ligase. So gene cloning is achieved then after a bacterium takes up the plasmid, and if the gene functions normally as expected, then the product, for example, insulin, may be retrieved. And here's a diagrammatic representation of that. So the polymerase chain reaction is a way of making billions of copies of a segment of DNA in a test tube. So PCR, it involves 3 steps that are repeated many times in cycles. First of all, denaturation, or denaturation, I guess you would say. The DNA is heated to 95 degrees C, and then it becomes single-stranded. Also annealing, the temperature is lowered then to 50 to 60 degrees C, and primers are added that base pair to the DNA to be copied. And then the extension at 72 degrees C, DNA polymerase used for PCR adds nucleotides to the ends of the primers, and eventually both DNA strands are copied and new stranded DNA forms. And this is a necessary process in order to actually do analysis of DNA samples, because quite often the DNA sample is very small, and so it has to be replicated many times in order to get a usable DNA sample. So the polymerase chain reaction is a chain reaction because the DNA is repeatedly copied. So the amount of DNA then doubles with each cycle. And then, so it has numerous applications, as mentioned. It includes identification of people based on their DNA fingerprint. Short tandem repeat profiling identifies individuals according to how many repeats of a DNA sequence he or she has at a particular short tandem repeat locus. And you can see an example of that there. So that ends this particular topic, and here's a summary of that we just looked at in terms of DNA cloning, and a summary of the key concepts that we just covered.